For millennia, extreme human longevity was confined to mythology. Today, advancements in artificial intelligence and genetics allow researchers to categorize aging as a solvable engineering problem rather than a biological impossibility. Global average life expectancy has grown drastically, jumping from roughly 30 years in antiquity to over 70 years today. We achieved this through sanitation, vaccines, and antibiotics. Yet the absolute maximum limit of the human body has stalled. The ultimate biological benchmark sits at 122 years. That is the age reached by a French woman named Jeanne Calment, setting a hard biological ceiling that modern medicine has yet to surpass. This graph illustrates the Gompertz-Makeham law of mortality. It shows that human mortality risk doesn't just increase steadily as we get older. It stays relatively flat for decades before it shoots exponentially upward once we cross the age of 80. Modern medicine is largely reactive. Doctors treat cardiovascular disease or cancer as they arise, applying interventions that make only tiny, insignificant dents in this steep upward curve. Standard treatments are mathematically incapable of breaking this exponential wall. Overcoming it requires biomedical interventions that address the base-level damage repair of the human machine. Until the late 20th century, scientists largely viewed aging as an inevitable entropic process, simple wear and tear on our biology. We now know that lifespan is highly malleable and actively regulated by our genetics. In 1993, a researcher named Cynthia Kenyon ran an experiment on microscopic nematode worms that proved lifespan could be intentionally controlled. By modifying a single gene known as DAF2, Kenyon doubled the worm's lifespan. These modified worms remained active and youthful far longer than wild-type worms. This table tracks how genetic pathways dictate longevity across species. By inhibiting nutrient-sensing pathways, researchers force biology to outlast its programming. In mammalian models, administering Senolytics to mice cleared inflammation, increasing their healthy lifespan by 36%. Aging is governed by genetic pathways. Because it functions like a biological software program, the code can be targeted, interrupted, and rewritten. To rewrite that code, researchers rely on a framework known as the Hallmarks of Aging. These hallmarks categorize the fundamental mechanisms of cellular breakdown that anti-aging interventions must reverse. One major hallmark is epigenetic alteration. Over time, the software that tells our cells which genes to turn on and off becomes corrupted. The cells lose their specific identity and stop functioning properly. Another hallmark is cellular senescence. Normal cells that sustain severe damage can turn into what researchers call zombie cells. They stop dividing, refuse to die, and secrete highly inflammatory molecules that degrade the surrounding healthy tissue. To counter this, scientists developed Senolytics. These drugs are engineered to selectively induce death in senescent zombie cells. They purge the toxic inflammation while leaving the healthy neighboring cells completely unharmed. For epigenetic damage, a separate approach is being built on the discoveries of Nobel laureate Dr. Shinya Yamanaka. He identified 4 transcription factors that, when applied to an adult cell, physically wipe clean the age-related epigenetic noise. This process reverts a damaged, differentiated cell back into a youthful, pluripotent stem cell. By combining these biotechnologies, medicine transitions from treating symptoms to systematically repairing the root hallmarks of biological decay. Attempting to override millions of years of evolution introduces severe biological roadblocks. The most prominent roadblock is known as the cancer paradox. The biological mechanisms required to promote extreme longevity frequently cross paths with the mechanisms that cause lethal tumors. This visualization shows the precarious biological balance researchers face. Extending telomeres or administering Yamanaka factors forces cells to live longer and regenerate. But unchecked cell division is the literal definition of cancer. Bypassing this antagonistic pleiotropy requires multi-system biological engineering. Researchers are deploying AI drug discovery models to design new molecules that can edit DNA and clear aggregates with absolute precision. Overdosing a rejuvenation therapy rapidly forms teratomas in animal models. Without flawless, non-toxic precision, a cure for aging becomes a lethal trigger for cancer. The biological and ethical hurdles are immense, and these systemic rejuvenation biotechnologies may not advance fast enough to achieve extreme longevity for people living today. Some turn to cryonics as a technological bridge. By cooling legally dead patients to liquid nitrogen temperatures in a process called vitrification, biological time is suspended. The premise relies entirely on future, ultra-advanced nanotechnology being capable of reviving the patient and repairing the original cause of death. Even with a regenerated heart or liver, a 1,000-year-old brain faces a physical limit. Neurons generally do not divide, and replacing them with stem cells risks erasing personality and consciousness. Furthermore, a biological organ has a finite physical capacity for a millennium of individual memories. Despite these bottlenecks, biogerontologist Aubrey de Grey calculates a timeline that suggests we are nearing the final mathematical threshold. He defines this threshold as longevity escape velocity. This is a specific tipping point where advancing biotechnology adds more than 1 year to human life expectancy for every calendar year that actually passes. Once medical science continuously outpaces biological decay, life expectancy effectively becomes infinite. If we reach that mathematical state in the coming decades, the first human to live 1,000 years has already been born.